Hello guys and welcome to another video of the chemistry syllabus. Today we're going to talk about post-translational modification and possibly go on to the next topic, which is physical properties of proteins. So post-translational modification. Why do we need it? First of all, polypeptide chains have primary, secondary, and tertiary and also quaternary structures, but sometimes you need to modify them after translation in order to achieve some biological functions, which we're going to talk about later. So the first type of post-translational modification is acetylation. So acetylation is the addition of an acetyl group. Acetyl group, what does that mean? It means CH3COOH. Okay, so it's a carboxylic acid with only one methyl group. The way we acquire the acetyl group is from acetyl coenzyme A. You're going to see this quite a lot in biochemistry, where you're transferring the acetyl group from acetyl coenzyme A to another molecule. And you do this, you add the acetyl group to the N-terminal of the amino acid chain. If you remember, N-terminal is the part with the free amino group or the very first amino acid. And so the enzyme that you need for this reaction to happen is N-acetyl transferase. N, because at the N-terminal, acetyl group transferase, transferring from acetyl coenzyme A to the amino acid chain. Right? So this enzyme is also loosely associated with ribosomes, so you will see it at ribosomes because that's where translation occurs. So acetylation, because we're talking about carboxylic acid, it can react with the amino group of the amino acid and therefore form an amide bond. Okay, it's not a peptide, it's an amide this time. Okay, but also sometimes you'll have side chains with an NH or NH2. Okay, so it's basic. Okay, and what can happen then is you can form another amide bond there, and so you can form multiple. There's multiple parts of the amino acid to form the acetyl or the amide bond between the acetyl and the amino group. Okay, so when you do this, the amide bond is pretty stable, so you cannot really accept another proton, and therefore it loses its positive charge. Now on to acylation. Acylation is the addition of a fatty acid to a polypeptide. Okay, also at the N-terminal. Fatty acids also contain a carboxylic acid group, and therefore they're able to react with the amino group of the amino acid or the side chain, or also if you have an oxygen or a sulfur in the structure, you can get ether or thioether bonds. For example, in cysteine, serine, threonine, stuff like that. The two main amino acids that we usually see is palmitic and myristic acids. And one interesting thing about the covalent bond between the fatty acids and the proteins or the polypeptides is that the fatty acids usually act as an anchor for the protein in the biological membrane. Okay, and you're going to learn more about this in the second part of biochemistry. Okay, so the third type of post-translational modification is hydroxylation. Hydroxylation is the addition of a hydroxyl group. Okay, so in this case, it's oxidizing an amino acid by adding an OH group. Okay, because it's oxidation by electrons, not by the addition of oxygen, because you also have a hydrogen there. Okay, the main amino acids to be hydroxylized are proline, lysine, and phenylalanine. And how you do that is you first of all, the enzyme required for this reaction is, you guessed it, hydroxylases. So sometimes you can usually figure out the enzymes used for a certain reaction by the name of the reaction, pretty much. So hydroxylation, hydroxylases, acetylation, and acetyl transferase, and so on. The hydroxyl group is added to the C terminal, to the C atom. okay, and that can be either the gamma or the beta carbon. And you can see, wait, I don't think it's labeled here, but yeah, you can see you can add the acetal group to this carbon or this carbon. So that's the gamma and the beta carbon atoms, and what you will get is if you oxidize hydroxyproline, you'll get proline, you'll get hydroxyproline. And an advantage of hydroxylizing amino acids is that you can later form hydrogen bonds, okay? So between the hydroxyl groups of hydroxylated amino acids, you can form hydrogen bonds which form cross links and therefore give structural stability. So like stuff like collagen, it's very important to have this structural stability, okay? Yeah, an important thing to keep in mind is that you also need other conditions like having oxygen and vitamin C in order to oxidize, for example, lysine, okay? And also NADPH, because you need to get the hydrogen from somewhere if you already have the oxygen. All right, so moving on, we have phosphorylation. So phosphorylation, probably one of the most famous post-translational modifications, and it acts as an on and off switch. It's an addition of a phosphate group, or PO4. Okay, so they're very reactive, and you can get it. You can add the phosphate group to the side chains of different amino acids or reactive amino acids. The enzymes that are used in order to phosphorylate, so that's to add a phosphate group, are kinases, whereas the ones that you need to dephosphorylate a polypeptide is phosphatase. Okay, so yeah, kinases for addition of phosphate groups and phosphatases for the removal of phosphate groups. And there's really no rule for kinases and phosphatases, because some of them activate polypeptides or enzymes, whereas others deactivate them. Okay, so you can activate or deactivate by the addition or removal of a phosphate group. Okay, so the addition of a phosphate group usually activates or deactivates a protein or an enzyme by changing its tertiary structure slightly so that it fits the substrate more, or doesn't fit the substrate or inhibit sort of the bonds between the temporary bonds between the substrate and the enzyme. Okay, so they cause conformational changes. And a good thing about phosphorylation is that it's very accessible in the cell. You can see it during almost all types of second messenger reactions. So cyclic AMP eventually activates kinases, which then phosphorylases some enzymes, which activates or deactivates them. And what starts all of this is the binding of a signaling molecule, a hormone like epinephrine, for example, to a G protein. Okay, so another post-translational modification to talk about is methylation. Methylation is the addition of a methyl group, which is CH3, and yeah, you can usually get this methyl group from transferring it from other methyl-containing substrates like fatty acids, for example. Okay, and the usual donor of methyl group is S-adenosyl methionine. So this is I think it's a nucleotide, and it's used for obtaining this methyl group. A lot of proteins are activated by methylation, but a big example of these are histones, calmodulins, and cytochrome C, which is important in oxidative phosphorylation. You're going to learn about that later. Okay, so moving on, carboxylation. Carboxylation is also another type of post-translational modification. From the name, you can guess that it's the addition of a carboxyl group. Okay, so a COOH. The main proteins that require carboxylation in order to activate them are blood clotting cascades. So that's like stuff like fibrinogen, prothrombins, stuff like that, clotting factors 2, 10, and so on. Yeah, so these proteins, they need to be carboxylated in the liver. Okay, so bear in mind this, we're still not at the site of clotting yet, of blood clotting yet. No, we're at the liver, where the activation occurs, okay, by an enzyme called alpha glutamyl carboxylase. Okay, and for this reaction you need vitamin K as a cofactor. Okay. Yeah. So here's the reaction. Oh. There we go, much better. So the second to last type of post-translational modification here is glycosylation, and this is basically the addition of a glycosyl group, okay, or a sugar or an oligosaccharide to the protein or the polypeptide. Okay, so this can happen. First of all, it happens in the rough endoplasmic reticulum, okay, which is where this primary is the site of primary protein modification, you know, and it can happen in two places: the N terminal, so therefore N linked, or O linked at the O terminal, okay. And what you get is you get an oligosaccharide. So if you remember when we were talking about sugars, oligosaccharide is basically a sugar conjugated with a polypeptide chain. Okay, and these are important because they form something called glycans, which are very important when you're talking about collagen formation and a lot of metabolic processes. But they also form glycoproteins, okay, which are receptors, okay, no receptor, signaling molecules, and basically they're like at the site of the biological membrane. You can see that they're specific to each cell type, okay, or organelle, and it's very interesting stuff actually. It all relates onto the biochemistry next semester, I think. So yeah, and then the last type of post-translational modification is glycation. So this is a bad type. Of course, there's no good or bad post-translational modification, but yeah, this is a bad type because this produces unstable glycosyl amines, or something called Schiff's base. Okay, and why these are not good is because eventually they cause the production of something called advanced glycation end products, or AGEs. So from the name age, you can tell that it has to do with age, which is pretty funny. I think they did that on purpose. But yeah, pretty much you can by figuring out the concentration of these advanced glycation end products in the blood, you can find out that they also affect other metabolic processes related to aging. Okay, and yeah, they're not good for the body because they disrupt a lot of metabolic processes, and they cause aging pretty much, you know, alongside oxidizing agents in the blood or in the body pretty much. Okay, so moving on to my more favorite part. Now we're going to talk about physiochemical properties of proteins, solubility and denaturation. So first of all, solubility of globular proteins. Specifically here we're talking about globular proteins because fibrous proteins tend to be mostly insoluble. Okay, globular proteins, if you remember, hydrophilic amino acids on the outside and hydrophobic amino acids on the inside due to a process called folding. Okay, so one thing to know about globular proteins, these by the way, these include albumins and globulins and stuff like that, if you remember from electrophoresis. But one thing to know about them is that they're pretty large if you compare them to other molecules like water or inorganic ions, for example. And what that does is it tends to form a colloid solution. So a colloid solution is essentially a kind of advanced solution where instead of the solvent molecules, you will have colloids, or sort of large, you can even, I think you can almost see them sometimes, molecules of proteins, okay, or large sort of, don't know how to explain. You'll have to see it, but if you've ever looked at a gel or something like that, you're probably looking at a colloid solution, okay. And yeah, it's because you're essentially enclosing the hydrophobic amino acids, and due to the large size of the molecule, you're forming these almost droplets in a solution, okay. But an interesting thing about colloid solutions is that you can dissolve them, but you can also separate them from a solution, okay. So one thing to talk about is salting in and salting out. Salting in and salting out. You've probably heard that word a lot, these words a lot, and you've probably also seen them in practicals. So yeah, salting in is basically using inorganic ions from salts in order to dissolve a certain protein or globular protein that is usually non-dissolvable. Okay, so let's say if you have a globulin, especially gamma globulins, and you add sodium chloride or table salt, you will see that these, now that you first of all, in normal conditions, the alpha globulins are not soluble in water because they form dipoles. And what dipoles mean is that you have a positive end and a negative end, and these attract, and they almost form like a long chain of magnets and long structures of magnets, and that's not good because then you cannot dissolve them, okay. But if you want to dissolve them, you can add inorganic ions which form a separation between these dipoles in the globular proteins of globulins, and then then they can dissolve. Okay, similarly, you can also use salt to aggregate certain proteins or coagulate them. And what that means is you're sort of taking them out of the solution. You're making a coagulant, okay, or a precipitate, okay. So for this one, you're going to need more saturated solution of salt, and you're also going to need inorganic ions that attract water more than these dipoles, okay. It's going to be tough because these dipoles tend to have very strong interactions, okay. So you need them to interact with each other and get closer to each other to form a precipitate. Okay, so now that we've talked about salting in and salting out, just the concept, we haven't really dived into any calculations yet. Okay, so as we were talking, globulins tend to have dipoles or dipole moments, and these interact with each other to form very hard to dissolve protein solutions. However, albumines, you don't really have these dipoles, and the charge is distributed more equally, therefore making it easier to dissolve and harder to aggregate or take or salt out, okay. So therefore you need a very high concentration of salt in order to salt out albumines. You need a fully concentrated solution of salt. So yeah, how you dissolve these globulins or albumines or whatever is you have a sort of envelope of water molecules around the proteins that act as a sort of shield, okay, to prevent them interacting with each other, okay. So we all know that water is a polar molecule. They have a slightly positive and a slightly negative end, and yeah, water molecules line up on the proteins in order to separate molecules from each other and form a colloid solution, okay. So a useful salting out agent, if you remember the concept of salting out, is ammonium sulfate, because in this salt the inorganic ions are very attracted to the water molecules, and therefore even more attracted than the proteins, and therefore they steal the hydration envelope from these proteins and force them to coagulate to make a precipitate. However, this surprisingly doesn't affect the activity of the protein. So if you were to dissolve them again, they would work just fine. They would catalyze the reactions, and yeah, you would still have the tertiary structure intact. Okay. Yeah. So now we on to talk about. I don't know why I'm a bit slow today, but yeah, now we're talking about pH. So what's the pH effect on coagulation? So first of all, thinking about it, I really like this chart or this drawing, because it shows you pretty much all there is to it. Of course, if you remember, the isoelectric point is the point at which the amino acids forms Witterions, and the protein has 0 overall charge. Okay, so if you have if you have 0 overall charge, then you have a positive and negative end. You have Zwitterions, you have the carboxylic acids forming negative parts and the amino groups forming the positive parts. You're going to find that you're forming dipoles. Okay, you're forming very reactive molecules, because then they can interact with each other, and they attract pretty much because you have the positive and negative ends. Okay. However, if you go away from the isoelectric point, then if you decrease the pH, for example, below the isoelectric point, you will see that the molecules tend to be positively charged, because the amino acid is accepting all the protons and becoming positively charged. Okay, so like we know, like charges repel. Okay, so therefore positive, if all the proteins are going to be positively charged overall, then they're going to repel, and therefore they're more likely to be attacked by the slightly negative parts of the water molecule and dissolve. Okay, so if you increase or decrease the pH below or above the isoelectric point, as long as you're not at the isoelectric point, you will see that the solubility increases. So it forms this graph. Whoa! I need to get a new stand. Okay, there we go. Please stay still. Okay, I'm just gonna hold it. All right, so on to osmosis. So this is where it starts to get slightly more complicated and slightly more about physics. So osmosis is basically diffusion of water. So in order to understand osmosis, we need to understand diffusion. So diffusion is the movement of molecules from a higher concentration, from a higher from the higher water content to the lower water content, okay? Or the movement of molecules from higher concentration to lower concentration. So if, for example, if you have here a lot of molecules, okay, and here you have no molecules, and this is a semi-permeable membrane, you will see that these molecules are like, well, we need to balance it out, because balancing out is the natural state of almost everything, and it needs to reach equilibrium. So it will keep going, the molecules will keep going back and forth until the concentration on either side is almost equal. Okay, the way to understanding osmosis is the same concept but with water. So for example, if you have more water on the right than on the left, okay, or more water content, so you have sort of molecule which is unable to cross this semi-permeable membrane, but the water can, okay. So what the water will do is it will say, oh well, there's less water here and more sort of molecules here, so I'm going to move in order to achieve equilibrium, and it's going to do that, okay, until the water gradient is balanced out, okay. And that's osmosis. Okay, so you have something called osmotic pressure, and this is the driving force of the water molecules to sort of diffuse to the other side, okay, in order to achieve equilibrium, okay. And you can counteract this this osmotic pressure by hydrostatic pressure. So if you remember hydrostatic pressure, for people who like physics, it's pretty much the pressure that increases as the water column weight increases. Okay, well, it increases, yeah, as the water weight column increases. So if you increase the volume, there's going to be higher hydrostatic pressure. Okay, that's how you can balance out this osmotic pressure. So the equation for osmotic pressure you can see here. Okay, osmotic pressure equals the difference in concentration times by the gas constant, which is 8.314, I think, and temperature in Kelvin. So this is, of course, reliant on temperature, because temperature gives water molecules kinetic energy, which allow it to move across a semi-permeable membrane more effectively, okay, and achieve equilibrium faster. Okay, so what's a hypertonic and a hypotonic and an isotonic solution? So hypertonic, hyper, meaning that it has a lot of salt, or a lot of, not salt, but it has a high concentration of molecules and a low water gradient, okay, and therefore the water will, no, okay, let's think about it this way. You have a cell, okay, and the cell is surrounded by a semi-permeable membrane, which it is, the biological membrane. If the solution outside the cell has a higher concentration of molecules than inside the cell, then the water will move from the high water gradient, which is inside the cell, to the lower, to where there's less water and more molecules on the outside of the cell. Okay, so that's a hypertonic solution, and that causes cells to shrink. Hypotonic solution, meaning that it lacks molecules, and therefore it has a higher water gradient. Okay, and therefore water will move from the outside of the cell to the inside of the cell, causing the cell to swell because now it's fuller of water. An isotonic solution is where the concentration of molecules is equal outside the cell to inside the cell, and therefore there is no net movement of water. Okay. All right. So, yeah, one thing to think about is the number of molecules. So you can have a molecule that will dissociate into two ions. Okay. So, for example, 1 mole of sodium chloride will form a sodium ions and chloride ions. So you will get 2 ions from 1 molecule. However, with glucose, you will get 1 molecule from 1 molecule. Okay, because there's 1 glucose. It doesn't dissociate any further. Okay, so 1 mole of sodium chloride is going to be to have twice as much osmotic pressure compared to 1 mole of glucose, because you have 2 ions. Okay, if I had 4 ions, it would be 4 times, and so on. Okay, so now let's sort of manipulate it more and think about it in a different way. What if we had 2 solutions, okay, separated by a semi-permeable membrane? However, on one side you have a molecule that can cross this semi-permeable membrane, but the other molecule cannot cross this semi-permeable membrane. Okay, what happens then? How do we achieve equilibrium? Okay, if all molecules are charged, of course. Okay, so let's draw this down and think of equilibrium that way. Okay, but before we get to that, the definition of gibbs-donnan equilibrium, okay, is basically what we were just talking about. So it's the equilibrium achieved when 2 solutions are separated by a semi-permeable membrane, where one solution has non-penetrating molecules, whereas the other solution has penetrating molecules. Okay, and this basically, what ends up happening is you'll end up getting an electric potential. So you will see an electric potential gradient. So you will see that the charge on this side, of the membrane, is going to be different to that side. Okay, which reminds us of neurons and pretty much how neurons work. All right, so here is the situation that we were talking about. You have these large yellow non-penetrating molecules, okay, and they're negative, and they're also attached to positive sodium or penetrating molecules. And on the other side, you have NaCl, okay, where you have positive sodium ions and negative chloride ions. Okay, so the sodium and the chloride ions can move across the membrane, but this molecule cannot. Okay, so you end up having higher concentration of chloride here than you have on this side. And for the chloride, we can expect it to diffuse until equilibrium is reached. When does it stop? How much diffuses? Okay, when is equilibrium reached? And how much sodium diffuses as well in order to counteract this chloride? Well, if you think about it, if in order to balance out the electrochemical potential, if chloride moves in, then the same amount of sodium ions from this side is also going to move in order to achieve electropotential equilibrium. Okay, so you need to balance out the charges, of course. Okay, so the equation for this equilibrium is, as you can see here. We're going to explain this in an example later, well, right now, pretty much, and you're going to see pretty much how it works. Okay, so first of all, let's call the protein molecules, negatively charged protein molecules, P-. Okay, so if we have a concentration of 10 millimoles of Na P-, okay, separated by a semi-permeable membrane, so the protein cannot cross, but the sodium and the chloride can. Okay, and on the other side, the other solution 2 here, will have 20 millimoles of sodium chloride. Okay, so what do we do then? All right, so first of all, we need to think about the direction of diffusion. Okay, so you have 20 millimoles of sodium chloride here, and 10 millimoles of sodium protein on this side. Okay, so we know that the concentration of chlorine, for example, is going to be higher on that side than it is on that side, on this side than it is on that side. Okay, so therefore we can expect chlorine to diffuse, and because chlorine diffuses, the same amount of sodium ions is also going to diffuse. Okay, so that's the direction of diffusion, and now we need to calculate the number of the concentration at equilibrium. Okay, so like we said, we can look back at the equation, and x in this case here, x represents the number of ions that diffuse through the membrane. Okay, so this is how many ions diffuse through the membrane, or how many the concentration of ions that diffuses through the membrane in order to achieve equilibrium. Okay, so first off, we start with C2 squared, C2 being the concentration of sodium chloride at equilibrium, divided by C1 plus 2 C2, C1 being the concentration of the sodium protein at the start. Okay, so first of all, we know that the concentration of protein is going to be 10 millimoles. Okay, because protein is impermeable, so the protein is non-penetrating. The protein is non-penetrating, so the concentration here is always going to be the same, and we already know that it's 10 millimoles per 10 millimoles. Okay, now for the sodium, this is where it gets tricky, because you have the sodium that's already bound to the protein, so that's 10 millimoles, plus the sodium that's going to diffuse following the diffusion of chloride ions. Okay, so after the chloride ions diffuse, you're going to get the same amount of sodium ions diffusing across in order to balance out the charges. All right, so yeah, you will have 10 millimoles plus x being the number of molecules of sodium that diffuse across, and what we're trying to find. And you're also going to have the chloride. You have no chloride ions to start off on this side, so the number of chloride ions that are going to diffuse is going to be x. Okay, or the concentration of chloride ions is going to be diffuse is going to be x. What is that sound? Okay, so and then we're going to also have the concentration of sodium. This is on the left side, by the way. On the right side, we're going to have the concentration of sodium, which we know that it starts at 20 millimoles, but you're going to have x diffuse. You're going to have x molecules diffuse from right to left. Okay, so it's going to be 20 millimoles minus x being the number of sodium ions diffusing following the chloride ions, and chloride is going to be 20 millimoles minus x being the chloride ions that end up diffusing to the left. okay? And then we can plug that into an equation, all right? And what you're going to get is, this is more complicated than it should be really, but this is a quadratic form of the equation, and then you can simplify that to get 50x equals 400, okay? So yeah, you can take this to the other side, or you can simplify the quadratic equation, which I don't know why they've done here, because this is really unnecessary, but if you want to do it that way, you can. All right, so, oh wait, they haven't simplified the quadratic equation, or if they have, they didn't show the steps. Let's stop talking maths and get back to chemistry for now. So now that we know what Gibbs-donnan equilibrium is, and we know the equation and how to use it, or we have a solid example of how to use it, we can start talking about oncotic pressure. So oncotic pressure is basically the pressure that is produced by the protein molecules of blood plasma. So as we know, these are non-penetrating, but they cause water to be diffused, or molecules to be diffused to their side, because they're non-penetrating, and therefore you need to achieve equilibrium by moving molecules from the other side to the side of the high molecular weight, or the non-penetrating proteins, okay? And that causes a net water movement, and therefore something called oncotic pressure, okay, similar to osmotic pressure, or the concept of osmotic pressure, okay? So the blood oncotic pressure, which is blood pressure pretty much, is caused by albumins, okay? So albumins are present in the interstitial, in the blood, okay? And because they have a high molecular weight and they're at constant movement, so there's always a concentration gradient, you're going to see that there's water being moved with, of course, with the other ions and so on, being moved from the interstitial fluid of tissue to the blood because of the albumins, because of these albumins cause the system to be at constant disequilibrium, okay? If it doesn't make sense, slow down the video and try to listen to it again and relate it to the parts before. I know I'm speaking fast, but I'm trying to keep it below 40 minutes. So yeah, so when you have low albumins, what you're going to see is you're going to have low oncotic pressure, and therefore low blood pressure, okay, which is a sign of hypoalbuminemia. So yeah, right. So that's that part. Now we're going to talk about dialysis. Dialysis works in the same way, okay. I'm going to unfortunately skip this part, and I'll advise you to read on it yourself. It's pretty much the same concept, but like I said, I'm still trying to keep this video under 40 minutes. Yeah, so we're talking about denaturation, and the last part of this unit. So denaturation is basically the disruption of the tertiary structure of the proteins. The tertiary structure of the proteins is basically the interaction between the domains and the polypeptide and the peptide chain within itself, so the way it coils after forming superhelixes or beta-cleated sheets. All right, and this structure, this tertiary structure, is very important because this way of folding usually forms the sort of shape for the lock and key hypothesis. Okay, so for enzymes to work, they need their tertiary structure. They need a certain way to fold in order to interact with their cofactors and in order to bind to their respective substrates. Okay, so denaturation pretty much means the disruption of tertiary structure, and this tertiary structure is kept together by multiple things like hydrogen bonds. Okay, so hydrogen bonds occur between the carbonyl groups and hydrogen atoms of the amino group, or even the side chains as well sometimes. Or you can also have hydrophobic interactions. So this is the dipole interactions between long hydrocarbon chains, okay, or hydrophobic parts. So basically, yeah, lipid solubility. And you're also going to have ionic bonds. So this is the parts where you will have side chains with basic or acidic parts will interact to form ionic bridges. Okay, and you also have disulfide bridges. So these are the only covalent bonds that keep the tertiary structure together. And what these do is they form cross bridges between cysteine molecules because they have sulfur. Okay, and these sulfurs tend to interact. Okay, so what causes denaturation? What affects the tertiary structure? So like we said, the tertiary structure is kept together by these interactions and these bonds. So you need to disrupt them, okay, in order to disrupt the structure. Okay, you need to, once you break these bonds that keep the tertiary structure together, the structure will start to fall apart. And you can do that in many ways. Heat. So when you heat something, you cause the increase of kinetic energy of molecules and of electrons as well, and they don't want to stay in a bond anymore. And if you heat anything, the hydrogen bonds are going to be disrupted. And yeah, hydrophobic attractions as well. The molecules are going to start to move more. They don't want to bind. They want to just move because they have high kinetic energy, and you don't get stable bonds. Okay. Ultraviolet radiation causes pretty much the same thing to heat by increasing the kinetic energy as heat, because it's pretty much the same thing. It's infrared radiation and then ultraviolet radiation. It still contains energy, photons of energy. Yeah, we talked about that in absorption spectroscopy, actually. A very interesting concept. And then you have strong acids and strong bases. So strong acids and bases interact with the hydrogen bonds, and they also saturate the ionic interactions. So you will see if you have a strong acid compared to the weak acid of the amino of the weak carboxylic acid of the amino group, you're going to see that it sort of substitutes this bond and forms another bond of its own. Urea as well disrupts hydrogen bonds. Inorganic solvents like ethanol, stuff like that, also saturates the hydrogen bonds and affects hydrophobic interactions. And then you have detergents like the ones that we talked about in electrophoresis and chromatography, and also heavy metal ions and mechanical aggregation. So if you mix it around too much, pretty much, yeah, and it also gives it too much kinetic energy, and therefore the hydrogen bonds get disrupted. So that's it for today. It's a bit of a long one today, but thank you so much for staying patient. I hope it makes sense, and if it doesn't, then go back and watch it one more time. You do have the book as well, so you can have a read through that. I'm going to try and do the protocols, but like I said, I'm in the exam period myself, so yeah, I'll do my best. Thanks again for watching, and I hope this helps. I really hope so. All right, you guys have a good night.